So in this video, I'm going to give you an overview of the process of respiration, which of course is the process that gives us energy to do things in our cells. So you probably know that there are two types of respiration. So there is aerobic and anaerobic. So aerobic respiration is glucose as our energy source plus oxygen gives us carbon dioxide and water and energy. And if you balance the equation, actually that's 6 oxygens, 6 carbon dioxides and 6 H2Os. And energy, of course, what we really mean in a biochemical sense is the molecule ATP. So there's another video all about ATP as an energy source in the cell that I recommend that you go and have a look at to understand why ATP is such an important molecule. There is also anaerobic respiration, so this is respiration without oxygen. So here we'd have glucose is converted, so no oxygen involved, to lactate or lactic acid plus energy. There are other forms of anaerobic respiration. It's not always lactate, but I'll use lactate as the most obvious example. And again, that energy, we mean ATP. So respiration is quite a complicated process. There's lots and lots of different reactions, but they all rely on the principle of coupling reactions. So again, a couple of my other videos do talk about this, but just to remind you what a coupling reaction is. So if we imagine on this page that any reaction going down the page is a favourable reaction in terms of its thermodynamics, and anything going up the page is unfavourable. Okay, so in a coupling reaction what you have is this. So let's say we've got two separate reactions. So we've got A goes to B, that's favourable, that releases energy, so that reaction is fine. But let's imagine in the cell we also want reaction C to D to happen. So D might be a really important thing in the cell. We need to be able to make D, but unfortunately it's an unfavourable reaction, so that is not going to happen unless we have an input of energy from somewhere. But what cells are able to do is to couple reactions together. So rather than having two separate reactions, what we have is this instead. So A goes to B at the same time as C goes to D, catalysed by some sort of enzyme. So that is the coupled reaction, where we're using the energy released from A to power the process C to D. Okay, so those are the fundamental principles of respiration. So let's have a look at what this looks like inside the cell. So here I've got my cell. If I just draw this out nice and big. Okay, so there's my cell. So let's start off in the cytosol. Okay, the first process that we need to think about. So respiration is divided into three processes. So the first process is the process of glycolysis. So glyco is sugar and lysis means breaking apart. So this first pathway, we start with glucose as our sugar. That's a 6-carbon sugar. And what we're going to do is to break up that glucose into smaller molecules. So that's why glycolysis gets its name. So there are quite a lot of steps in glycolysis. And I really don't want you to be worrying about all of the individual steps. You'll probably find other videos that go through it step by step, all of the biochemistry. That's not what I really want you to focus on. I want you to focus on some of the overall principles. I will go through the first reaction. So the first reaction, the very first reaction that has, is we go from glucose to glucose 6 phosphate. So we add a phosphate group to the glucose molecule. So we go from a 6-carbon molecule to a 6-carbon 1-phosphate molecule. And if you've already watched the ATP video, you will know that this is an example of a coupled reaction. So this reaction this is an unfavourable reaction. This is an example of a C to D type reaction. So that won't happen unless we put in some energy. So we actually put in the very first thing we do, which might seem really bizarre, in a process that's supposed to make ATP, the very first thing we do is to spend some ATP. We actually use up some ATP. The energy from that reaction is used to power it, and actually that phosphate there that we've added to the glucose is the phosphate that was released. Remember, ATP has 3 phosphates, ADP has 2 phosphates. So we've added the phosphate to the glucose molecule, and we've used the energy released from that to catalyse that. And that is catalysed by an enzyme called hexokinase. So hexokinase, hexo again is 6, kinase is an enzyme that adds a phosphate. So this enzyme has added a phosphate onto glucose using ATP for both of its energy and for the phosphate itself. So there we have an example of a coupled reaction. That's one of the classic examples. Here's actually the very first reaction of glycolysis itself. So we've then got a whole bunch of other reactions. As I say, I'm not going to go into each and every one of these reactions, but the most important thing is the next important product that we get to is something called fructose 1,6-bisphosphate. So that is a 6-carbon 2-phosphate molecule. So glucose and fructose are both 6-carbon sugars. Just the carbons have been rearranged a little bit. And we've added, you'll probably notice, another phosphate to the molecule. So again, we've used up some more ATP. So at the moment, this process has cost us 2 molecules of ATP. But we will get there in the end. The next thing that happens is that molecule splits to give 2 molecules of triose phosphate. So each of those molecules is 3 carbons and 1 phosphate. So we've split it into 2 molecules. So everything that happens from here on in happens twice because we've got 2 molecules of triose phosphate that comes from our glucose. So the molecule splits. There is then a series of what I often call the boring reactions to get down to a molecule called pyruvate, which is a 3-carbon molecule. So we've gone from 6 carbons to 3 carbons. I really don't care about all of the intermediate molecules in here, but what I do care about is, so you'll notice that we've got rid of phosphate, so we actually make back 2 molecules of ATP. But remember, we were actually doing this twice, so we've now got 4 molecules of ATP that we've made. We spent 2 to start with, so net is 2 ATP per glucose. Okay, so although we used some ATP to start with, we've already got it back. I remember all of this happens twice because we've got 2 triose phosphates. And then the other thing that we get back at the end of this is a molecule called NADH. And that is another energy carrying molecule. Okay, so we synthesize this energy carrying molecule at the end of the process. So net for glycolysis, we've got 2 molecules of ATP and we've got 1 NADH. In fact, that's not true. We'll have 2 NADHs per glucose because this bottom bit's happening twice. So we get to pyruvate, which is a 3-carbon molecule. There's 2 different things that can happen here. let's deal with anaerobic respiration first. So one of the things that can happen is if there's no oxygen, then what will happen to pyruvate is it can be converted to a molecule called lactate or lactic acid. And in doing so, we can squeeze a little bit more out of it because we can make some NAD+. So basically what's happening is reversing there. If we have all of the NADH, all the NAD+ has been used up, then we can't make NADH and we can't get any more energy out of it. So what this does is to break down some of that NADH. That means this whole cycle can go and we can still get those last couple of ATP molecules out. But lactate is a metabolic dead end. The only really thing that can really happen to lactate is it can be converted back to pyruvate. So we go back, NAD+, NADH. So that is anaerobic respiration. So we don't get any more ATP from the system, and we have to do this sort of weird, funny little reaction to keep any ATP going. So anaerobic respiration is really not very efficient. So you actually only get 2 ATPs per glucose molecule. And that's not really very much because we're not really using the whole potential of the glucose molecule. So now we're going to move on to aerobic respiration, and you probably know that to do aerobic respiration we need to go into the mitochondria. So let's draw just one massive mitochondrion on my piece of paper. Okay, and you probably know that mitochondria have a double membrane. Just going to show, and I'm just going to put the inner membrane coming across there, which will be important in a minute. Okay, so this is the mitochondrial outer membrane. That's the inner membrane. So the space in between them is the intermembrane space, and this would be the matrix, which is sort of the equivalent of the cytosol in the mitochondria. Okay, so we've got our pyruvate. Pyruvate, if we're going to do aerobic respiration, we move in to the mitochondria, and that's get converted to a molecule called acetyl-CoA, which is it's actually not just a 2-carbon molecule, it's 2 carbons attached to a big carrier, but in terms of what's important, there's 2 carbons there. Okay. We've gone 3 carbons to 2 carbons, so we release our first molecule of carbon dioxide. So we're starting to get that equation balancing there. There's the CO2. It's not all of the CO2, it's some of it. And that gives us some more NADH. Okay, which is, as you remember, is another energy carrier. So once we've got acetyl-CoA, we can go into a process that you may have heard of before, which is the Krebs cycle. It's also called the citric acid cycle or the TCA cycle. It's the same thing. Okay. And again, you'll probably find other videos on the internet that will go through all of the reactions in the Krebs cycle. I don't want you to be thinking about all of the individual reactions. What I want you to know about is the first reaction is a molecule called oxaloacetate, which is a 4-carbon molecule. 4 carbons, we take the 2 from acetyl-CoA, so that goes in there, to make citrate, which is 6 carbons. So that's why it's called the citric acid cycle, is because we make citrate as the first important product of it. There's then a whole load of different processes. I'm just going to summarise them. I do not want you learning all of those if you don't want to. Those molecules get converted between each other to regenerate oxaloacetate. To do that, we have 2 molecules of carbon dioxide get released. So we use 1 carbon dioxide here, 2 carbon dioxides here. So that's all of the carbon in the pyruvate is now converted to CO2. So we've got no carbon left. But what we have made along the way are 3 lots of NADH. So NADH is an energy carrier. We make 3 molecules of that. We make 2 molecules, just 1 molecule of another carrier called FADH2. Sorry, I'll just write that there. We make 3 times NADH, 1 times FADH2. So those are carrier molecules. And we make 1 molecule of GTP. So we go from GDP to GTP, and that GTP can become ATP. So we've got another ATP in as a result of that. Okay, so these are all energy carrier molecules. So where are they going to carry the energy to? Well, most of the energy, so ATP can then just go off and do whatever it needs to do. But this NADH in particular needs to go to another process which is happening at the inner membrane of the mitochondria. So we have a bunch of proteins in the mitochondrial inner membrane, which are referred to as an electron transport chain. So again, I'm not going to go into all the details of each and every protein in here. What I'm looking at is the overall big picture stuff. So we've got loads of NADH. That were a product of the Krebs cycle. NADH can release its electrons to make NAD+. So NADH is an electron carrier. It can give up its electrons to become NAD, and that releases loads and loads of energy. That's minus 206 kilojoules per mole. That's loads of energy. Those electrons kind of get passed between one protein, one carrier and another in a series of quite complicated steps that you don't need to know, don't need to worry about too much. But eventually that those electrons are given to oxygen, and in doing so we make water. So that's where the oxygen is coming from in our aerobic respiration equation. So you might be like, well, what's the point of that? Why are we doing that? Well, the reason that we do that is to use the energy that's associated with moving these electrons around to move protons across the membrane. So we generate a proton gradient whereby in the matrix we have a relatively low concentration of protons, but in the intermembrane space we have a high concentration of protons. So we pump protons across the membrane. It's got really acidic space in this intermembrane space. And then if you've watched the ATP synthesis video, you will know that we use that proton gradient to make our final amount of ATP. So the ATP synthase sits in the membrane. It's quite a complicated looking enzyme. Looks something like that. So there's a bit in the membrane that actually rotates, and then there's a head group that sits on the matrix side. So what happens is those protons go back through the enzyme. Because they're moving from high concentration to low concentration, that's favourable, that's releasing energy, and we can use that energy to finally make shed loads of ATP. So we did make a little bit of ATP along the way. So we made some in glycolysis, we made some in the Krebs cycle, but most of it comes from this electron transport chain and ATP synthesis mechanism. So with all of that, so it is really complicated. There's lots of little steps in there. But as a result of that, going through all of the chemistry, instead of just getting 2 molecules of ATP out, we can get 32 molecules of ATP out if we do aerobic respiration. So it's obviously, if you can do that, then it's a good idea not to stop at pyruvate and lactate, but to take things into the mitochondrion, go to acetyl-CoA, go through the Krebs cycle, burn all the carbon to CO2, make loads and loads of this NADH molecule, use the energy from the electrons to pump the protons, and then move the movement of the protons finally to make ATP at the ATP synthase complex. So there's quite a lot going on there. And if you look at it in textbooks, then they will go through all of the individual reactions for you. As I say, I think what's important is not the individual carbon compounds. You'll notice I've only written out 1, 2, 3, 4, 5, 6, 7, 8 carbon compounds. There's about 20 in there in total. What's more important is where is it happening? So which organelle is it happening in? Is it making or consuming ATP? Are we making or consuming NADH? And again, all that energy going towards the electron transport chain to pump the protons to use in the ATP synthase complex. So as I say, there's a video that talks about this process of ATP synthase a little bit in more detail, and there's another video that sort of talks a bit more about the coupling reactions and ATP. But that's been a whistle-stop tour through the process of respiration.